Bismillah ar-Rahman ar-Rahim. Bismillah ar-Rahman ar-Rahim. The subject of the title of our lecture is hypersensitivity, and the objective of this lecture is type 2 and type 3 hypersensitivity, and the mechanism of these hypersensitivity diseases, diagnosis, and the treatment. The hypersensitivity, also called the hypersensitivity reaction or intolerance, and refers to undesirable reaction which is produced by the normal immune system, including allergies and autoimmunity. So it is a normal immune response against the pathogen or against the antigen, but there is a damage to the normal tissue. This leads to affect the function of that organ which is affected by the immune system. And the best example is allergy and the autoimmunity. And normally the immune response is removed by the antigen by various mechanisms without extensive damage to the host tissue. While under certain circumstances, this inflammatory response can have a harmful effect, resulting in significant tissue damage, and this inappropriate immune response is termed hypersensitivity. Hypersensitivity is the word implies an increased immune response, but instead it's an inappropriate immune response to an antigen. Hypersensitivity occurs in the course of both humoral or cellular immunity. And the two scientists, Gill and Coombs, proposed a classification in which hypersensitivity reactions are divided into 4 types. So, type 1 hypersensitivity, which is mediated by the IgE. The important antibody in this type is IgE. And the type 2 hypersensitivity, which is the main mechanism, it's antibody-mediated cytotoxicity. While type 3, it is immune complex-mediated hypersensitivity. And type 4, which is delayed type hypersensitivity reaction. And there is a 5th type, which is occurred due to an autoimmune disease, which is receptor-mediated, like Graves' disease, myasthenia gravis, and the main important antibody is is IgM or IgG, with association with the complement function. Now we will deal with type 2 hypersensitivity, which is the main mechanism. It's antibody-mediated cytotoxic hypersensitivity, and the reaction involved the antibody IgM or IgG. This leads to destruction of the normal cells by 3 methods. First, the complement activation and force formation, which is the antigen-antibody immune complex, lead to activation to the classical pathway of the complement and lead to formation of force on the cell membrane due to MAC formation, membrane attack complex, which is the end result of the complement activation. And the second mechanism, antibody-dependent cell-mediated cytotoxicity, that the cell did not destroy the target cell or antigen or pathogen unless the presence of the antibody. And the third type, which is antibody serve as an opsonine enabling phagocytosis that increase or enhance the phagocytic activity of the cell with the Fc or C3b receptors that bind the binding occurs through these receptors, and then phagocytosis occur. The best example is transfusion reaction of incompatible blood, and secondly drugs like penicillin, and hemolytic disease of the newborn. The first example is the transfusion reaction of incompatible blood. Here the RBCs had an antigen, large number of proteins and glycoproteins, which are considered as an antigen on a cell surface that encoded by different genes, and these known as ABO blood groups. And this is considered as antigen when they are transfused to another person. And the antibodies will form against these antigens are known as isohemagglutinin, which is the characteristic of the IgM, which is the isotype IgM, as you know previously, the IgM has a characteristic for heme agglutination. If a type A blood group individual is transfused with a blood type B, so there is an immediate reaction, and it's called immediate transfusion reaction, occurs in which the anti-B isoheme agglutinin IgM bind to the transfused RBCs of the blood group B and mediate their destruction. And this is the mechanism is complement-mediated lysis, and this is occurred intravascularly. And this reaction occurs within hours after a blood transfusion, leads to free hemoglobin in the plasma due to destruction of the RBCs intravascular. This is very important, destruction of the RBC intravascular. And this hemoglobin filtered through the kidney, and this filtered through the kidney, resulting in hemoglobinuria, and there is clotting within the blood vessels. Symptoms: the patient complain from fever, chills, nausea, pain in the lower back due to involvement of the kidney. And treatment: first, termination of the blood transfusion immediately, and to the patient diuretic to get rid from the free hemoglobin in the blood. The second type of transfusion reaction, which of incompatible blood, which is the RBCs had a large number of proteins and glycoproteins on the cell surface. And the second type of the incompatible blood transfusion, which is the delayed type, and this is generally occurs in individuals who have received repeated blood transfusion of ABO compatible blood, like in patients with thalassemia or leukemia or renal failure had repeated blood transfusion. It's compatible with the ABO blood group, while which is the major blood group, while incompatible for the minor blood group like KIT, RH Russell system, C, D, and E. As you know, we did only RH for the patients. And there is another minor blood groups, kill DOPI, and this reaction occurs between 2 to 6 days after blood transfusion. The mechanism here, stimulation of the IgG antibody production against these minor blood groups, and the RBCs will be destroyed extravascular, while in the immediate type intravascular, while here extravascular by agglutination. This is the mechanism, by agglutination, opsonization, and phagocytosis by the macrophage, while in the immediate type the mechanism is complement-mediated lysis. And the patients will complain from fever, jaundice, and anemia. So the patient had repeated blood transfusion and still anemic, and this is due to delayed type hypersensitivity reaction of the compatible blood transfusion. It's compatible to the ABO, while incompatible to the minor blood group. Another example is hemolytic disease of the newborn, and you must concentrate and pay attention. It is hemolytic and not hemorrhagic. There is hemorrhagic disease of the newborn, and here the hemolytic disease of the newborn. And this is developed when maternal IgG antibody is specific for the fetal blood group, antigen RH process system, CDE, across the placenta and destroy fetal RBC. And most commonly develop when the father is RH positive and the mother is RH negative, that do not express the D antigen on the RH positive fetus, express an RH antigen D on its RBCs. And this is occur, as we said, in the RH incompatible female. Here the fetus, the mother is RH negative and the father is RH positive, and the fetus, same to his father, RH positive. So the RBCs had, the RH positive or RHD positive antigen on the fetal RBCs. So the first baby is not affected, and during the delivery of the first baby, during the delivery of the first baby and separation of the placenta, so there is some of the fetal RBCs across the placenta and go to the maternal circulation, cross the placenta and go to the maternal circulation. And this lead to stimulate, this is the fetal RBC, lead to stimulate the B cell of the mother. And the B cell will be differentiated into the plasma cell and memory B cells. The plasma cell will secrete the IgM. This is 5 immunoglobulins will secrete the IgM. IgM, the characteristic did not cross the placenta, so the first baby is not affected. While in the second baby, when it is Rh positive also as his father, and during delivery and transmission of the fetal RBC, which is Rh positive, and transmitted through the placenta and go to the mother circulation. And this lead to stimulate the memory B cell. And this memory B cell will differentiate into a plasma cell. And this secrete the IgG because it is secondary immune response. This is the first or the primary immune response which secreted IgM, and here the secondary immune response and secreted IgG. And the main character of the IgG across the placenta. So this immunoglobulin IgG will cross the placenta and go to destroy the fetal RBC. And there is destruction of the fetal RBC. So to prevent this, we will give the mother ROGA, the anti-D, which is so the mother treated with ROGA. This is the B cell, and this is the ROGA, which is antibody against the antigen, which is present on the cell surface of the RBC, and this leads to neutralization of all antigen which is present on the RBCs. So prevention, antibody given to Rh negative mother within 72 hours after delivery of Rh positive baby, and anti-D given to Rh negative woman in 28 weeks of gestation. They found that this is not sufficient after 72, so give the mother first dose after when she was in 28 weeks of gestation, and the second one after 72 weeks of gestation. And also we give the mother after abortion, because we did not know the blood group of aborted fetus. And usually mothers are injected with the anti-D antibody at about 28 weeks of gestation, as mentioned here. And the second injection occurs at about 34 weeks, and the last dose of anti-D given after delivery within the 72 hours after delivery. So the mother have 3 doses of the anti-D, 28 weeks of gestation, 34 weeks of gestation, and 72 weeks of the gestation. So the mother have 3 doses after 28 weeks, 34 weeks, 72 hours after delivery. And to detect this disease, we use direct Coombs test. In order to diagnose this disease, we send the patients to direct Coombs test. And here the patient sample RBCs coated with antibodies, and then we add to it anti-IgG, which is Coombs reagent, called Coombs reagent. This reagent is anti-IgG, and this leads to